All right, ladies and gentlemen, in this presentation, we're going to discuss two separate things. First is transport of atoms and molecules in the universe, and then specifically the cell membrane and how the two things relate. So the first thing that we want to talk about is a real fundamental concept that is incredibly important to nature as a whole. Atoms and molecules and compounds are constantly in motion. The air around you is constantly in motion. Water in a water bottle or a swimming pool or a sink is constantly in motion. Molecules you can and cannot see are constantly in motion. And when they are in motion, they are always moving to spread out from where they are. And they will continue to spread out until they reach equilibrium. Take a look at this diagram over here. So you can see here you are dropping a liquid that is colored into water, presumably. And you'll notice that at the start, the drop is collected all together. All those little round circles are molecules from that drop of color. And what you'll see is as time moves forward, the drops start to spread out from each other. And they spread out from each other until you can see in this last spot, they're all equally spread out. And that's what really happens. Eventually, molecules will spread out until they reach an equilibrium. Generally, this means that they never stop moving. They'll just start moving slightly, but they will keep moving until they get as far apart from each other as they can. Now, the most important thing about this is this happens because that's the natural state of the universe. Atoms and molecules always want to spread out. They do that because that's the lowest energy state. That's how nature wants to be. Now, when you have two substances or more mixed like this, so we have our little colored substance here and our water here, what we have is what's known as a solution. All right? Now, a solution is a liquid mixture of multiple compounds or multiple atoms. And a solution always has two parts. The first part right here is the solute, and that's the substance mixed in. So in our example here, that would be the colored substance coming out of the dropper. And then you also always have the solvent, which is the substance the solute is mixed into, and that, in this case, would be our container of water. Water is generally the solvent for all living things. All right, so water is the solvent for living things. So we always talk about living things and substances in living things in terms of how they react with water. And that's going to be a big deal as we get into the structure of the cell membrane. And the last big deal I want to talk about here is this idea of concentration. That's a term that we're going to use a lot. And concentration is basically the ratio of solute to solvent. How much solute do you have to how much solvent do you have? And you could measure that in milliliters, you could measure that in grams per milliliter, you could measure that in percentages, but it's always a measure of how much of any given substance you have, and its ratio to the solvent. Now, those are some basic concepts of how atoms move, all right? What we want to talk about now is the structure of the cell membrane. So the cell membrane is an organelle. It's an organelle that all cells have. Eukaryotic, prokaryotic, animal cells, plant cells, bacteria cells, fungi cells. All cells have a cell membrane. And the cell membrane is made of lipids. So we talked about how in living things, lipids are one of our 4 organic macromolecules. And they're a very important organic macromolecule. And they are responsible mostly for energy storage. But one of the other main jobs that they do is they create the main structure of the cell membrane. And the reason that they create the main structure is because they have two parts. There is always a part of a lipid up here which loves water. And there is always a part of a lipid down here which hates water, a phospholipid. And these phospholipids can use this to create a barrier so that you can definitively have inside and outside of your cells. Having phospholipids like this allows you to compartmentalize life. And as soon as you can create compartments, you can start to create unique structures. All right. So let's first talk about what this phospholipid is. You will constantly see it drawn like this with a little circle that's the hydrophilic head and these tails that are the hydrophobic part. All right. But what's actually going on here is you have this structure right here, which is a phosphate group, which is charged. Okay. You have this glycerol, which is kind of like almost like a collar, and the phosphate group and the tails are attached to it. So it's really just kind of a placeholder. And then down here, you have fatty acids. And if you recall, fatty acids are monomers of lipids that can combine to make triglycerides. You'll notice from the diagram that fatty acids can be saturated and straight, or they can be unsaturated and have a kink. But depending on which gives the phospholipid slightly different properties. All right, but both are possible in it. So what happens with these phospholipids is they create a structure like what we have here. All right. So this is what we call a phospholipid bilayer. And here we have the outside of a cell, and here we have the inside of the cell. And this part of the lipid bilayer and this part of the lipid bilayer love water. They want to mix with water. They sit with water. They mix with the solvent water. That's why we say they're hydrophilic. But then what you have inside here with all these tails is a hydrophobic portion. Those fatty acid tails, the fatty acids are lipids. Lipids are not going to ever mix with water. And so what this does is this creates a situation where things that will mix with water hang out over here. Things that will mix with water hang out over here. But things that mix with water can't get through the middle. And so the bilayer becomes a barrier to entry. And that's what makes up the majority of the cell membrane. But the cell membrane is not just a phospholipid bilayer. The cell membrane is what we call semi-permeable. That means some things get in and some things get out. And the cell membrane has to be able to control what gets in and what gets out. And the way that it does that control is through the properties of the phospholipid bilayer, so that deals with things that are lipid-like and things that are not, but also by using proteins. Okay? So, you will notice here on this diagram, this is actually what any given section of any membrane on any organelle in a cell will look like. It'll have phospholipid bilayer, and then just sort of stuck inside of it, you'll have all these proteins, carbohydrates on some of them, you'll have cholesterol, all right? And this is what makes up what we refer to as the fluid mosaic model. A mosaic is a type of painting where you take like individual colored tiles and you assemble them together, and then when you look at them from far away, they kind of look like something. And so we call it this way because all of these pieces in the membrane are like those little tiles. And when you look at them individually, they're like, oh, okay, that's what it does. But when you back up and look at them at the big picture, they are what controls what gets in and out of the cell. All right, that's why we call it the mosaic model. We call it fluid because this stuff is all moving throughout the membrane. It's constantly changing position and altering. And that actually gives the membrane some very unique abilities in terms of letting things in and out and controlling cell structure and stuff. Okay? So, let's talk a little bit about some of the stuff that makes this up. For starters, we're going to go with cholesterol. Now, I'm sure you've heard about cholesterol. Cholesterol is bad for you. It gives you heart disease. Except that cholesterol, like everything that's potentially bad for you, serves a purpose. And the purpose of cholesterol is to keep your cell membrane fluid. It actually creates a situation where it keeps the tails from getting too close to each other, which keeps it fluid. And it also kind of acts like an anchor so that the tails can't spread apart too much, which keeps it fluid. And it actually allows them to help the membrane resist temperature change. So it actually allows the membrane to stay fluid when it's cold and when it's hot. So cholesterol does play an important role. It's bad for you because you're able to make all that you need, so eating it is not a good thing. Now, We've already talked about the phospholipid bilayer, how you have the heads, the tails, they form the majority of this. The inside is not water soluble, the outside is. And what you also have are these proteins. Now, proteins can do two things. They can either span the entire membrane, which means parts of it are on the outside, parts of it are on the inside. And when that happens, we refer to them as integral, because they span the entire membrane. Or you can have proteins that just sit on one side, and we call those proteins peripheral. Okay, so there's peripheral proteins and there's integral proteins. Now, these different proteins can do all kinds of different jobs. You see up here, this is called a glycoprotein. That's because it has glycogen on it, the carbohydrate. Those types of proteins are used as ID tags for the cell. So the cell basically can say to your body, hey, this is me, I'm this kind of cell, there you go. And you could see that over here as well. All right. Now, these right here are a very important kind of protein because they create a gap in the phospholipid bilayer. And these proteins are unique because they only allow certain things to pass through. So these transport integral proteins are one of the structures that makes the membrane semi-permeable, that allows it to decide what gets in and what gets out. Now you also have receptor proteins like this, where something binds to the outside. This changes shape, and then something happens in here to like all this cytoskeleton that's attached, and that sends signals throughout the cell. All right? So there's a lot of different types of proteins that sit inside the membrane. They all do different jobs. The membrane is made mostly of phospholipid bilayer, and cholesterol helps keep the membrane fluid. And you'll notice that since cholesterol is inside the tails, cholesterol is lipid soluble, not water soluble. So cholesterol is a lipid. All right, so that's part one on our presentation on the cell membrane and transport.